Hi everyone, so today I wanted to talk a little bit more about the laboratory setting for the diabetic condition. And that's to say, what are researchers and scientists doing in the labs to imitate the diabetic condition for which all of our research around diabetes is based? And I wanted to do this for a couple of reasons. The first is because when we as humans hear about diabetic research, whether it be on animals or humans, the thought is always taken for granted that, they're doing all of these things in mice or humans that are diabetic. But we aren't really thinking about, well, how do you make a mouse diabetic? Or how are you controlling the diabetic state in these mice? Are you breeding diabetes in mice? Are you inducing diabetes in mice? And then for humans, what are the mechanisms of, or what are the variables that you're paying attention to in diabetes? And animals are far more common in the research because they are, for lack of a better word, disposable, and that's not really a nice way to talk about them. But it's a lot easier to go through generations of mice. They're euthanized, and then their conditions are analyzed. Now, because there is so much research done in these mice, how are they making them diabetic? And if you look further into how you induce diabetes, my logic was, well, if you know how to create diabetes, then why wouldn't you know how to not create, like to undo what you've done? And again, it's not one-to-one, it's not a perfect lineup, but at least to give you an understanding of when they do go on to talk about what treatment they ended up doing in whatever paper it is you're looking at, you're able to understand, okay, if it's something that's anti-inflammatory, well, what about the diabetic condition was tied to inflammation from the beginning? You know what I mean? So that's one, because to learn how it's created in the lab, you may learn inadvertently how to kind of reverse engineer it a little bit, or at least understand the variables that are being controlled for the research. The second reason that I would be interested in it is because the animal model of diabetes and the ensuing results, there's been a lot of really cool research talking about beneficial treatments for mice with diabetes. But then when it goes to apply to humans, it falls flat. And in all of my experience of, I find an exciting paper and I bring it to my doctor, and the doctor, looks at it and says, that's cool, but I did notice it's in an animal model. There's not a lot of crossover between animals and humans. And that's so frustrating because why? Okay, that's fine. Why are we doing all of this research in animals knowing that there isn't a strong correlation with how it turns out in humans? That's a little frustrating. But also, one thing that I consider a lot about is, what about the animal diabetes is different than our diabetes? And I would argue that the human diabetes is far more intricate and complex in terms of the organ systems that are being involved. So, for example, and we're going to talk about this later in the video as well in the research when we talk about these specific substances that are used to induce diabetes. But basically in animals, they're injected with a substance that specifically targets these, specifically targets the beta cells, right? So you inject this, beta cells die off. It is inflammation based. So toxic substances targeted for beta cells based on the inner workings, inner proteins of those beta cells, and they die off. That is not how it develops in humans, right? It is far more complex, involving a lot more pieces, right? So for humans, for example, liver health. Liver health is so important in balancing the body as a whole and inadvertently influencing the health of the pancreas and beta cells. Does any research in any sort of diabetic model in animals account for liver health? No. And the substances that they're injecting in these mice, it's not affecting the things that are affected in the human diabetic condition, like the liver, like the kidneys, like our gut health. Now, those variables in isolation are sometimes treated in mice. That makes it the topic of the paper that you're reading, right? So if you're reading about gut health and diabetes, there's, they say, well, we manipulated the gut of this mouse and saw what it did for its diabetes. With humans, like, the gut comes before the diabetes, right? These are mice who have been made diabetic chemically and then had their gut microbiome changed. Humans have just been this petri dish since their birth of all of these different things that make things a little bit more complicated. Does that make sense? So now let's go ahead. We're gonna switch over and we're gonna start to look at some of the mechanisms for creating diabetes in the animal model, so that way we can know where these papers are coming from. And when it comes time to draw correlations between what we're finding in animals and how we can apply it in humans, we have a better understanding of where that animal model is coming from, so that we, learning and knowing more and more about the human variables that go into the development of diabetes, we can apply it more aptly and with better control.
So the two substances that are used to induce diabetes in the laboratory model are alloxin and streptozotocin. And this paper does an excellent job of breaking down the mechanisms of how each of these substances induce diabetes. So I'm going to start with alloxin. So alloxin has a couple different means of creating diabetes. The first is by interfering with proteins and enzymes that are responsible for regulating blood sugar. You'll see here it says glucokinase. So I brought this up over here. Just a quick little Google here. Glucokinase is a gene which plays an important role in recognizing how high the blood glucose is in the body. So glucokinase is something that is sensing the changes in your elevation of blood sugar after taking in glucose and initiating the chain or the cascade of reaction that ends up with the release of insulin. So looking back here, alloxin has the ability to inhibit glucokinase, right? So you can see here inhibition of glucokinase. Now, it also induces ROS, or reactive oxygen species, which we know is basically the currency of inflammation in the body. So this results in selective necrosis or death of beta cells. Now, an interesting feature of alloxin as well as streptozotocin is that they are glucose analogs, in that they look very similar to glucose. And a lot of the mechanisms for which glucose enters the body is also how alloxin gets into the body. You'll see here that the alloxin molecule is structurally so similar to glucose that the GLUT2 glucose transporter, which is one of the pathways that glucose enters the cell, in the beta cell plasma membrane accepts this glucomimetic and transports it into the cytosol. So this, insulin is so commonly called like a key that lets glucose into the cell. Alloxin is something that looks so similar to glucose that when insulin goes to let glucose in the cell, Alloxin is something that looks so similar, it just goes in right behind it. Now, in this section, we're talking about some of the specifics about how alloxin targets glucokinase, which is this. It's not necessarily a kingpin, but it's an important feature in the proper metabolism of glucose. And so, alloxin, you can see here, inhibits glucokinase, which in turn produces glucose oxidation and ATP generation. So the cell is unable to make energy because, again, that's how glucose works. Insulin lets glucose into the cell, which reacts with the mitochondria. Mitochondria generates energy. Now, an interesting point here is talking about how glutathione, which you are able to supplement and is a naturally occurring antioxidant within every cell. This mentions that glutathione, among several other of these thiols, protect glucokinase against the alloxin attack because they are able to interfere with the metabolism of alloxin, which is what ends up being toxic to the rest of the cell. In its ability to also create reactive oxygen species, once alloxin has been let into the cell through that GLUT2 transporter, it starts to get reduced into something called dialuric acid. Now, dialuric acid is something that initiates, you can see here, generates superoxide radicals. Superoxide radicals are a type of reactive oxygen species. So it creates these superoxide radicals and hydrogen peroxide. And then once those ROS are present, then comes the Fenton reaction, which I have talked about ad nauseum in my podcast. So the Fenton reaction is basically taking the less severe superoxide and hydrogen peroxide ROS and turning them into something far more volatile in the hydroxyl radical, which you'll see here. In the presence of a suitable metal catalyst, typically iron, hydroxyl radicals, okay? So alloxin gets into the cell, it's broken down into dialuric acid. Dialuric acid generates these superoxide radicals, which eventually decompose into hydroxyl radicals, which wreak havoc on the health of the cell. It goes on to mention a few of the ways that the diabetes that is induced by alloxin can be guarded against. One is an increase in glutathione, right? As well as something called superoxide dismutase, okay? These are both, even in the name, superoxide dismutase, superoxide, it's something that is breaking down superoxide and preventing it from decaying into hydroxyl radicals. Now, SOD and then GSH is the abbreviation for glutathione. The antioxidant enzymes are able, you can see by virtue of their ability to scavenge superoxide radicals. These are what prevent alloxin from being able to induce the damage that it does. But interestingly enough, the fact that dialuric acid makes superoxide, it's been shown here, they're referencing here, the superoxide radical is not the species responsible for the cytotoxicity of alloxin and dialuric acid. Several lines of evidence point to none other than the hydroxyl radicals as the principal culprit. Now, we talked about that before with the Fenton reaction. The Fenton reaction, including iron, is how those superoxide and hydrogen peroxide molecules are being degraded into these hydroxyl radicals, which are the ones that are causing the issues. Knowing that the creation of superoxide and hydrogen peroxide degrading into hydroxyl radicals is ultimately what is causing diabetes is pretty cool. They are now, they've spent the past few paragraphs here talking about how you can interrupt how those superoxide and hydroxyl radicals are converted to hydroxyl, right? Or not hydroxyl, you're taking the superoxide and the hydrogen peroxide and preventing it from becoming the hydroxyl. The hydroxyl is enemy number one, right? These are the ones, hydroxyl radicals interact with biological targets before they can be inactivated through hydroxyl radical scavengers. So hydroxyl radicals work so quickly and so violently that most of the means of interfering with them aren't fast enough. So this is going on to say a multitude of metal chelators, iron chelators in particular, because iron is the main culprit in how hydroxyl radicals are produced. But they're saying that the results obtained have not been unequivocal. So it's like sometimes, they work a little bit, but not really. Basically, they're saying, given the extremely short half-life and the extraordinary chemical reactivity of the hydroxyl radical, it is not surprising that complete protection against alloxin toxicity cannot be achieved, right? So it really doesn't matter what you're using to block hydroxyl radicals. They work so quickly and so violently, they're very, very hard to get in front of.
An important thing to note after this is that they talk about how glutathione is an effective means of guarding against this inflammation model of creating diabetes. So glutathione is a naturally occurring antioxidant that happens in typical cells, but it is also abundant in several foods, as well as being able to be supplemented. An important thing to note after this is that they talk about how glutathione is an effective means of guarding against this inflammation model of creating diabetes. So glutathione is a naturally occurring antioxidant that happens in typical cells, but it is also abundant in several foods, as well as being able to be supplemented. So with alloxin-induced diabetes, the alloxin is imitating glucose and a substance that is typically allowed into the cell. Once it is allowed into the cell, it starts to change what would typically happen when glucose was taken in. So alloxin is broken down into dialuric acid. Dialuric acid is responsible for the creation of superoxide, which is a reactive oxygen species, as well as hydrogen peroxide. So these two in and of themselves are not horrible ROS in terms of their effect on the cell. But what happens is that this dialuric acid is creating more and more ROS, which create this kind of pool of ROS that are liable to be converted into hydroxyl radicals by the Fenton reaction, which involves iron. Okay, so while what alloxin degrades into isn't the exact thing, it's basically putting fuel, like loading the cell up with fuel to allow for this extra iron to come in and light a fire that essentially burns the cell out.
Now, streptozotocin works very similarly to alloxin in that it is imitating a compound that is typically taken in. In this example, they talk about how streptozotocin is an analog of something that is typically taken up by a GLUT2 glucose transporter in the plasma membrane. So streptozotocin is basically like a toxic version of something that the GLUT2 glucose transporter, which is one of the ways in which glucose gets into the cell, recognizes and lets in. Now, on the question of why streptozotocin targets beta cells and how it's so damaging to them, is based on the nature of what streptozotocin is. It's something called a nitrosourea, which if you just give it a quick look up here, it'll say it's a DNA alkylating agent. So it's something that manipulates the expression of DNA. So looking back here, it's largely assumed that the toxicity of streptozotocin is dependent upon DNA alkylating activity of what it is. So when streptozotocin interacts with DNA molecules, it causes damage, which can result in the fragmentation of DNA. But it also messes with protein glycation, which affects basically what these proteins are able to do, something like insulin. And when this happens in the cell, it basically throws on the red alarm and tries to fix everything. And in that kind of like craze to save itself, it basically depletes its own energy and dies. So when they talk about addressing what the mechanism of this drug is, they talk about ways that they can prevent it. Nicotinamide is one of the things that they mention. Nicotinamide is another term for something called niacinamide. Niacinamide is a version of vitamin B3. Okay, so that's important to remember. So they were saying that nicotinamide, prior to the administration of streptozotocin, is a well-known protectant of beta cells against the toxic action of the drug. And just like with aloxin, they talk about how it may not be the major factor of streptozotocin, but it is a residual factor in that it is able to generate ROS, specifically superoxide and hydroxyl radicals, which are what are causing that inflammatory cascade.
And with streptozotocin, you're able to see that how it enters the cell by kind of mimicking something that is typically allowed in without any issue. It's just like aloxin, right? It's kind of like an imitation of a toxic imitator of something that the cell trusts. But once the streptozotocin is inside the cell, it's not generating the same reactive oxygen species that the aloxin is. Streptozotocin instead is alkylating DNA, which is means of altering the DNA structure. And when this happens, the cell is like, oh my God, the DNA is being messed with. Let's get the immune reactions fired up. They start to burn a lot of energy trying to correct the damage that's being done by the streptozotocin. And in doing that, it depletes its own energy sources. And because the DNA is now changed, it's unable to effectively continue to create glucose to fuel that immune reaction. They do talk briefly about some of the protective mechanisms that can be used to prevent the DNA alkylation. They mentioned nicotinamide, which is another name for niacinamide, which is a version of vitamin B3. So, food for thought, when you look at the fact that aloxin induces reactive oxygen species, which creates this hydroxyl radical, which just wreaks havoc on the cell, breaks things down, and causes cell death. And you compare that with how streptozotocin interferes with the DNA and how it engages the cell's repair mechanisms in a very energy intensive way that eventually leads to the cell's death because it's not able to create energy anymore because its DNA has been messed with. An interesting point that ties the two together is the fact that just looking at the mechanisms, so ROS creation is toxic to beta cells. DNA alkylation, DNA messing with the DNA of the beta cell causes a immune reaction, a repair mechanism that is harmful to the cell. How can we take each of those situations and apply them to human diabetes? Well, interesting enough, a high degree of inflammation like that caused by aloxin and creating all of this ROS and hydroxyl radicals is or has been shown one of the residual effects of elevated ROS and elevated hydroxyl radicals is DNA alkylation and changes to DNA structures. So the way I see it is that in creating those hydroxyl radicals with alloxin, that is showing the toxicity of the reactive oxygen species being caused in an environment where iron can react with it to create the hydroxyl radicals, which are the most dangerous ROS. Now, strepsizotocin is just one step downstream because it's saying, well, it doesn't create ROS itself, but it messes with the DNA. By messing with the DNA, it causes all of these repair mechanisms which harm the cell. The fact that it goes right to the DNA alkylation does nothing to tie it, like it, ROS leads to DNA damage. Okay, so alloxin is basically like step one, killing off the beta cells. Strepsitocin is just like getting in at step two. So yes, the ROS toxicity itself is enough to kill the beta cells, but also it also can create an environment where even more inflammatory conditions, even more DNA damage can be done downstream from the ROS itself.